Chapter 1 Molecules of Life Subtopic 1.5 Nucleic Acids Learning Outcomes. At the end of lecture, you must be able to state the structures of nucleotide as the monomer of nucleic acids, and you must be able to state the types of RNA. Nucleic acid is polynucleotide. It is a polymer of nucleotides, meaning many nucleotides are joined together to form a chain of nucleotides. The building blocks or the basic unit of the nucleic acid is nucleotide. Mostly it composed of carbon, hydrogen, oxygen, phosphorus, and nitrogen. There are two types of nucleic acids. Number 1, deoxyribonucleic acid, or also known as DNA. Number 2, ribonucleic acid, that also known as RNA. Structure of nucleotides. Nucleotide has three components. Number 1, pentose sugar. Number 2, nitrogenous base. Number 3, phosphate group. Pentose sugar can be divided into two, ribose and deoxyribose. Ribose can be found in RNA, whereas deoxyribose can be found in DNA. Pentose sugar is a 5-carbon sugars. From the diagram, you can see this is the carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. Pentose sugar can be divided into two. Deoxyribose, that can be found in DNA, ribose, that can be found in RNA. The difference between deoxyribose and ribose is at carbon number 2. In ribose, carbon number 2 has oxygen atom, whereas in deoxyribose sugar, carbon number 2 has no oxygen atom.
In ribose, the second carbon atom is linked to the hydroxyl, OH group, and a hydrogen atom, H. But in deoxyribose, which has one oxygen atom fewer, the second carbon atom is linked to two hydrogen atoms, H.
Another component of nucleotide is nitrogenous base. There are two groups of nitrogenous base. Number 1, pyrimidine. Number 2, purine. They are grouped based on the number of carbon ring. Pyrimidine has single carbon ring, whereas purine has double carbon rings. Nitrogenous base under pyrimidine are cytosine, uracil, and thymine, whereas under purine we have guanine and adenine. In RNA, we have nitrogenous base such as cytosine and uracil, guanine, and adenine, whereas in DNA, we have cytosine, thymine, guanine, and adenine. These are the diagrams of nitrogenous base. These are pyrimidines, and these are purines. Pyrimidine has single carbon ring, whereas purine has double carbon rings. Nitrogenous base in DNA are guanine, adenine, thymine, and cytosine, whereas nitrogenous base in RNA are guanine, adenine, uracil, and cytosine. This is DNA nucleotide. and this is RNA nucleotide. The components of nucleotide are nitrogenous base, pentose sugar, and phosphate group. Nitrogenous base join with pentose sugar at carbon number 1. Phosphate group join to the pentose sugar at carbon number 5. Formation of dinucleotide. When two nucleotides are combined by condensation process, it will produce dinucleotide. The nucleotides are joined together by phosphodiester bond. The phosphodiester bond is formed between hydroxyl group in phosphate group of one nucleotide and the hydroxyl group at the third carbon in pentose sugar of the other nucleotide. Formation of dinucleotide. This is one nucleotide and this is another nucleotide. The nucleotides undergo condensation process. During the condensation process, the hydroxyl group at the third carbon in pentose sugar of one nucleotide is released, and at the same time the hydroxyl group in the phosphate group of another nucleotide is also released. So during the condensation process, the water is released. The nucleotides are joined together by phosphodiester bond. Formation of polynucleotide. When many nucleotides join together to form a chain of nucleotides, it is known as polynucleotide. Polynucleotide elongates from 5' to 3' direction. The polynucleotide has a backbone with repeating sugar and phosphate units. Sugar, phosphate. Polynucleotide can be broken down into nucleotides by hydrolysis process. RNA. RNA is a single-stranded polynucleotide and it has ribose sugar, and the nitrogenous bases are guanine, adenine, cytosine, and uracil. There are 3 types of RNA. Number 1, messenger RNA, or also known as mRNA. Second, ribosomal RNA, or also known as rRNA. And the last one is transfer RNA or tRNA. The function of mRNA is to carry the genetic information copied from DNA, which act as a template for protein synthesis. The function of rRNA is to form ribosomal subunits during protein synthesis. The function of tRNA is to transfer specific amino acids to ribosome during protein synthesis. This is a diagram of tRNA, and the amino acid will attach to the attachment site on the tRNA, and the tRNA will transfer the amino acids to the ribosome during protein synthesis. Differences between DNA and RNA. DNA has 2 polynucleotide chains, whereas RNA has 1 polynucleotide chain. Pentose sugar of DNA is deoxyribose, whereas the RNA has ribose as a pentose sugar. The nitrogenous bases in the DNA are guanine, adenine, thymine, and cytosine, whereas the nitrogenous base in RNA are guanine, adenine, cytosine, and uracil. The DNA is a double helix, whereas RNA is linear. DNA has longer chain or larger molecular mass, whereas RNA has shorter chain or smaller molecular mass. DNA has only one type, whereas RNA has three types. There are mRNA, rRNA, and tRNA.